Giuseppe asked me to do is to not only show data from our lab, and in fact I'm going to be very light on data and from our lab, but kind of speculate a little bit about the future of vaccines. And the first two slides are my 2 cents on that. One is that I see that there is a real need for conquering major diseases that are chronic diseases. Here you see tuberculosis, hepatitis C, malaria, HIV. So going forward, these are real big targets for vaccine development in the next few years. And another big target that is going to be in the future of vaccines is constituted by emerging and re-emerging diseases, where there is a need for treatment and diagnostic and vaccines. And we heard in the first part of the morning quite a bit about that as well. So in terms of forward trends, there is a convergence of interest in fighting some of these targets by philanthropic organizations, the Gates and others, NIH and other funding agencies, and industry and biotech. And I think this is particularly true for San Diego, where a lot of these interactions occur, and this is a fertile ground for this type of interactions. And also, talking about emerging and re-emerging disease, if I think in the future, what's going to happen is that certainly they are here to stay. And it's actually interesting to me to see how damaging emerging diseases have been in the last few years. Just think about avian flu, the swine flu, SARS, and so forth. And really, there's a tremendous direct and indirect, especially impact. This has really not much to do necessarily with vaccine development of science, but also with we as a society. I think we have to find better ways to deal with emerging diseases without freaking out. And if you think about the damage that was made by SARS, which was very regrettable, but was somewhat limited as compared to the damage that was done to tourism industry, travel, and so forth. It's this indirect impact which is caused by information more than disease. And so, but that's I don't want to go too far off on the philosophy. And go back to what we do in a lab. And we follow some processes which are very akin to what has been commented earlier in terms of trying to understand what are the targets of immune responses and then use those, the knowledge derived by those analyses to maybe design vaccines or to assist in various different things, including the study of host pathogen interactions. So perhaps quantitate responses of vaccines, not as a response against the whole organism, but perhaps against that specific epitope that we are interested in. We are interested mostly in T-cell responses, and we used this process starting from the sequences of the antigen of interest, doing some computer analysis, and then predict a bunch of different epitopes that are then tested in the lab with different specimens from diseased individuals or people that have resolved the disease. So I'm going to give you 3 really quick snapshots. One has to do with smallpox, the other has to do with allergies, and the final has to do with TB. And apart from smallpox, the other 2 are very much preliminary data and work in progress. So there was a resurgence of interest in smallpox as a result of 9/11. There was a desire to develop new second-generation smallpox vaccines because of the safety problems associated with vaccinia virus, which is the current smallpox vaccine when that vaccine is administered to immunocompromised individuals, and also with the significant, albeit low, number of just reactions. So because all the work done with vaccinia was done a long, long time ago, virtually nothing was known about what were the exact targets of immune responses. And so we set out to do that, to define and try to understand what were the different targets of immune responses following vaccination with vaccinia virus. And apart from, so the main interest of that was to be able actually to compare the immune response at the fine level induced by different new second-generation vaccines, because of course, as you're aware, you can't really do field trials with smallpox in humans because there is no smallpox in the wild to assess efficacy. But that's all done and gone. But we were interested in understanding better what was going on with vaccinia virus, because vaccinia virus, it's a big thing, has more than 200 different ORFs. So we were curious what variables determine whether the immune system is going to recognize this or that protein. How is immunodominance regulated? And we were particularly interested in a bunch of different variables that had to do with, maybe it has to do with how much of this antigen is made, where it is made, when it's made, what's the stability, and so forth. And so one thing that we noted early on is that there were certain antigens that are immunoprevalent. So if you look at an immunomic map, if you wish, of all the different antigens that are recognized by, say, different strain of mice, of macaques, or humans, and so forth, there are certain antigens, this is for CD8 responses, that are recognized in the context of many different MHC types. So, for example, D1R is recognized as dominant antigens by, in the concept of KD, KB, DB, HLA-A1, A3, B7, B44. So there's something about this antigen that makes it likable or preferred for CD8 responses. And obviously that has nothing to do with the HLA allele or MHC allele. Also what we noted quite early on was that there was this same kind of pattern of immunoprevalence for CD4 responses, but the pattern was quite different from the antigens recognized by CD8 responses. Shane Crotty used a technique developed by Phil Feldner to map on a micro slide different vaccinia antigens expressed in a recombinant fashion and interrogate with this technology the serum from a mouse in this particular case, but the same experiment was done in humans, to light up what are the immunodominant antigens for antibody responses. And again, I'm not showing here the data, I'm just telling you what the result is, so that while CD4 and CD8 were focused on certain antigens and were focused on different set of antigens, antibody and CD4 responses tended to go hand in hand. There was not perfectly, but there was a significant association between targets from recognized by CD4 and antibody responses. So as I was mentioning, one thing that we're interested in is looking at the pattern of expression, both at the RNA and protein level. And we did that. We actually had to do this from scratch because it turned out there was no comprehensive study where the different ORs have been looked in parallel with the same assay the same day. And we defined 4 different classes of expression at the mRNA level for POX antigens. And bottom line is that what we found out is that the antigens that are preferentially recognized by CD8 responses are the ones that are made immediately, very early. Those are made abundantly in the dendritic cells that are infected, and they are also turned over very rapidly. They're not very stable. They're enzymes, and they end up in the class 1 pathway very efficiently. Dendritic cells are not very permissive for infection, complete infection by vaccinia virus. And in fact, what we see is that CD4 response and antibody responses tend to focus on structural component of the virus that are made late, and they enter the processing pathway through the exogenous pathway. So at that point, it's not important whether you're made early. It's important that you're made abundantly, and you're very stable because this component of the virion are very, very stable. This slide just summarizes what I was mentioning earlier on. And basically, here the bottom line is that we learned that there is this dichotomy of what the antigens that are recognized by CD8 and CD4 responsive antibodies, which also has relevance if you're trying to make vaccines that are going to boost in a prime boost mode. You need to be cognizant of that. What are you trying to boost? And the antigens you want to use are quite different. Now, moving gears for a second to the second part of the talk is that has to do with immunoallergone. This is a completely different story. NIH has given us a contract. It was their idea to map all the T cell epitopes from common allergens, and the idea here is to try to define T cell epitopes to make a second generation immunotherapy, if you want. You probably all are aware of the fact that there is a specific immunotherapy for allergies, the allergy shots, and those is based on a very careful gradual dose escalation of injecting people with ground up pollen or other extracts. And the reason it has to be done very slowly is that these are the same antigens that bind IgE, and so if you're not careful, you just give it all in a bolus, you're going to have a nice anaphylactic reaction in your hand. It's not really known exactly what happens, why people get better if you give them specific immunotherapy, but there is the general idea is that this is because you activate T cells that have somehow a regulatory effect and drive away a response from a Th2 type of response. So we studied this in Timothy Grass. There are 10 known allergens. We map all the epitopes and so forth, and the response is very heterogeneous. In these 10 different proteins, we map 43 different epitopes, and there is no need to go through the details. And the, right, I'm not going through it. But let me show you a couple of other pieces of data which I think are very, very intriguing. And this is what we learned as a side product of this fishing expedition. Very often you do something, you map something, when you realize that there is something very interesting coming out from the mapping study. And so in this particular case, so you see the response, the T cell response against the Timothy Grass extract, so this ground up pollen. And these are all overlapping peptides spanning through all the different 10 known allergens. And some people respond perfectly well to Timothy Grass, but don't see any of the peptides from any of the known allergens. So this suggests that you're kind of missing the boat here. There's something that is obviously recognized by T cells that is not the known allergens. And in fact, if you run a 2D gel from an allergic patient, there are plenty of other proteins that are recognized by IgG, not by IgE. You talk to the allergists, they say, oh no, we knew that. That doesn't matter. How does it matter? So because these are the known allergens that have different isoforms. So basically, from an allergy point of view, the only thing that matters are whatever antigens bind IgE, because those are the ones that induce a response. But there are many, many other proteins that are also seen by the immune response, except it's not a pathogenic response. And in fact, we now sequenced and made peptides from some of these proteins, and we know that a large fraction of the T cell responses in allergic people is directed against these allergens. And if you also compare people that have gone through specific immunotherapy, and you do the comparison here, it's not worth doing it right now, but there are plenty of proteins that appears to be specifically recognized in people that go through immunotherapy. So this coming from an infectious disease mindset, it's intriguing because that's the first thing I would be interested in. So wait a minute, you're giving someone a shot, they get better, but you ignore the proteins at which the immune response of the people that got better is directed in. And the answer from the allergologist, it doesn't matter. Those don't bind IgE. I know they don't bind IgE, but that's what I want to give to people, because if I was an allergic patient, I'd rather get the green stuff than the red stuff, because the red stuff is what makes me sick. So there you go. Now, moving to the third issue here, the third part of the presentation, tuberculosis. I don't need to elaborate on the fact that tuberculosis is bad. And also, you are probably aware of the fact that there is a lot of evidence that speaks to a role of T cells in the control of disease, such as the fact that the gold standard for diagnosis of TB is interferon gamma release from T cells, or the fact that immunocompromised people that are latently infected with TB reactivate. There are instances of spontaneous resolution, BCG, which induces a powerful T cell response, affords some protection, and so forth. And it's also, of course, data in animal models. Now, TB is made of 4,000 ores, actually a little bit over 4,000 ores. And yet, if you look through VIDP, for example, only very few of those proteins have been described as target of T cell recognition, or containing T cell epitopes recognized in humans. So now, this means that 5% of the genome is what's out there as being recognized by T cells. So either your immune system is seemingly blind or oblivious to 95% of the genome, or again, there's something that has not been looked at as yet. And so our working hypothesis is that there is a lot that has to be discovered yet. The genome of TB in reality has not been available for so long, and this is probably why truly genome-wide studies have not been performed. And also what we want to understand if there are differences between people that have a good outcome versus a bad outcome. So going back to the same things that were talked earlier in the morning, people that have neutralizing versus non-neutralizing here would be different to people that resolve versus non-resolve, people that become latent versus people that become active, either qualitative or quantitative differences in the response. So we've done a genome-wide screen. We synthesized 20,000 peptides, and Cecilia, which is in the audience, screened them all in gazillions of different cells and patients and stuff. So I'm not going to bore you with the data, but the point here is these are 1,000 different pools of 20 peptides each, and this is the frequency of recognition in latent individuals. And you have to take my word for it that many of these antigens that are prominently recognized in latent individuals and thereby have contained the infection are totally novel, have not been described before. So we're taking it to the next step now and doing comparative studies of disease, different disease cohorts, latent versus active, treated, household contacts, and also generalize that not only to this San Diego cohort, but to other cohorts from Brazil and hopefully from Peru and Los Angeles, which is the farthest away from San Diego, of course. And we're making tetramers, and because we know what the epitopes are, we can make tetramers, and we can really ask fine questions about what is the nature of the T cell responses, and are they qualitatively different in different disease outcomes. So conclusion, there is a lot more to the TB genome and its interaction with immune response that is known thus far. And our goal is to draw complete immunome mapping of host TB interaction as far as adaptive immunity goes. And also the precise definition of the epitopes and restriction element would hopefully allow to tease this one step further and determine whether there are qualitative differences in different disease outcomes. And that's it. like to, oh, I borrowed this from, I edited it, obviously I don't need to thank myself. Sorry about that. Egomaniac. But I'd like to thank all the people involved. Bjorn Peters and Jay Greenbaum have been involved in all the computational aspects and in general throughout this collaboration with Howard Gray, Denise. John Sidney has been doing all the MHC stuff. Shane has been a great collaborator throughout many of the studies, and specifically also the pox virus. Cecilia Lindestam has been doing all the TB work, and these are some of the many collaborators that have been helping with the patient recruitment, both in the allergy and TB. And thank you very much.